Okay, so we are here for a new spotlight interview with another PLOS ONE Lab Protocol article. In this case, we have Raven Osborn. Raven is a current PhD candidate in translation biomedical sciences with an emphasis in bioinformatics and immunology at the University of Rochester. Her current research interest is on using applications of network theory in conjunction with high-throughput transcriptomic and protein data to understand how SARS-CoV-2, the virus that causes COVID-19, evades the innate immune system. She grew up in Kansas City and spends her time outside of the lab advocating for diversity in STEM, hiking, skiing, rock climbing, playing kickball, doing trivia, cooking, and reading fiction. And we will be discussing, as I mentioned before, her PLOS ONE Lab Protocol paper, preparation of non-infectious scRNAseq samples from SARS-CoV-2 infected epithelial cells and the associated protocol in protocols.io. Raven, thank you so much for sparing some time out of your busy schedule to talk to us.
Thank you so much for having me. I'm so excited and thankful that, the protocols.io and PLOS ONE collaboration even exists. So thank you for having me.
So to begin with, can we hear from you what the protocol is about, how you use it, and why it is relevant?
Yeah, sure, absolutely. So essentially, the protocol is just step-by-step on how to isolate tough to dissociate epithelial cells that have been infected with SARS-CoV-2 and currently now, and at the time where I did it, then SARS-CoV-2 is still classified as the BSL-3 virus or pathogen. And we really wanted to use a live virus in single cell sequencing or any types of analysis that we wanted to do instead of using just the proteins. But we needed to get it out of the BSL-3 environment in order to do the sequencing. So it's the protocol that we created in order to get that done. And the reason why that's so important is because early in the pandemic, you would see very few single cell sequencing papers. And if you did see them, then they would be, paragraphs of authors. And the reason that would be is, because they would have to do it with, collaborating with, these huge institutions, or maybe they were at these, institutions that have lots of money and highly equipped BSL-3 facilities. But ours was, pretty minimally equipped at the time. I don't think anyone was doing any virus work at all. So we wanted to, make a low cost, method for us to actually use live virus instead of just, one protein, and just really be able to understand what's happening from an infection, after infection, and not just one molecule at a time. So, yeah.
So would you say then that the motivation for optimizing this protocol was to make research on SARS-CoV-2 more, not only affordable, but more accessible for universities that didn't have the high security facilities?
Absolutely. So the way that it's done is, even if you don't have a BSL-3 environment, if you can get the samples out and to somewhere that has sequencing, within like a few hours, then you should still have high quality samples, which was really important to us. So yes, definitely there are very few BSL-3 facilities in the United States. And for the most part, a lot of them are more like bacteria, and you don't have like these, these single cell sequencing platforms are tens of thousands of dollars. And a lot of the times when you have the facility, people tend to keep them minimally equipped because once something goes in there, it kind of has to stay. And maybe every 10 years or so, then you'll get like like a really good cleaning, but that costs tens of $1,000s to decontaminate the entire facility as well. So a lot of the time you have to be really smart about the equipment that you actually add into those facilities. Yeah.
And were there any published methods or protocols for doing what you wanted to do? And if that is the case, why you saw the necessity for optimizing and creating this new protocol that you published both in the platform and then had it peer-reviewed and published as a standalone methodological paper?
So I would say yes and no. There were, because we're working with SARS-CoV-2, there were a few inactivation methods that had been published for SARS-CoV-1, but they were just viral inactivation, meaning like this virus is no longer able to infect metabolically active cells, which is so that can just throwing like Trizol on them, like on the cells, something like that'll inactivate the virus. But for us, we needed single cell data, which means that the cells can't be lysed, and so we didn't see anything that had used the viral inactivation with single cell. I had done a literature review, and we found that paraformaldehyde and Trizol and methanol acetone would be able to inactivate the virus, but we don't know how those methods worked using it with the single cell platforms. Like, what does that data look like in the end? So we kind of combined methods, and once we sort of settled on something that we were hoping would work, and presented it to our institutional biosafety committee, obviously, then we optimized from there to make sure that the RNA that we're getting from the samples was at least comparable to what we could get without doing any of those inactivation methods.
Okay, thank you. And we started this conversation by you actually mentioning the partnership between Protocols.io and PLOS ONE. So my question is, what was the motivation to have your protocol peer-reviewed?
Well, we started this project. We had to get, oh my goodness, so many background checks in order to start doing the work. And so we probably started around the summer of 2020. And at that time, there were like a few, articles that came out like, okay, well, this is what we're doing here at the University of Rochester. And so people would, ask like, how are you guys doing this? Because we're creating all those methods all at the same time while we're getting the facility, ready to be used for SARS-CoV-2 work. And so people would ask, and we would, and since we had for the BSL-3 facility, everything has to be very detailed steps, and it had to be approved by the institutional biosafety committee, and the people who are working at our core facilities who do the sequencing, they all had to be comfortable with allowing us to do this, right? So we already had like we were creating a protocol and making it like shareable for other people who are interested in doing that type of work. And so we decided, instead of having to wait for someone find whatever papers that we come out with later, and then maybe reach out years later when, hopefully I'm I'm gone and done with my PhD, then why not just make it searchable? Part of us doing this is to make sure that it's accessible, and part of accessibility is being able to find it in the first place. So that's how we wanted to have it as a peer-reviewed article, as well as putting on protocols.io.
And can you comment on the feedback that you received from the reviewers from PLOS ONE, how their critique shaped the final version of your paper in this case?
What's interesting, I think that this was my first paper that I've ever written, and I was pleasantly surprised. I know a lot of people dread the peer review process, but I was pleasantly surprised. I believe that the reviewers were really excited about this type of protocol being out, because it is really important to have that accessibility. They did help me to think a little bit more about some of the applications that it could be used for that we hadn't tested yet. So for instance, one of the reviewers were asking if it could be used for other high throughput technologies like flow cytometry. And so after I thought about it and really looked into whether or not we could do this, because then obviously I would want to try it too. But I basically found that with the methanol acetone inactivation, then it really depended on the fluorophore. If it was protein-based fluorophores, then it wouldn't work as well unless you did a really, really thorough washing steps. Then it can sort of quench the signal from like APC. So I would say that types of feedback, really thinking critically about the other applications, was really helpful.
And is that something that you addressed experimentally, or that's something that you addressed in the discussion?
Oh, just in the discussion. Yeah. I would say most of the feedback that I got from the beginning was through our institute, our IBC, our Institutional Biosafety Committee, and the core facilities, because they wanted to be absolutely sure that we wouldn't be in the news for for a lab leak. So yeah, that was really important.
Okay, yeah, that totally makes sense.
Yeah.
And I would like us to dig a little bit more behind the scenes. And I don't know if while you were developing and optimizing this protocol, was there anything that you tried that didn't work and didn't make it to the final version of the protocol?
So I'll say that we had to change the way that some of the single cell sample processing was done compared to what they usually do in our core facilities. So for instance, I found that using methanol acetone to inactivate it, it made the signal from trypan blue. It affected it in a strange way so that you couldn't tell which cells were alive or dead. So I was gonna put that into the published protocol, but I didn't find that it was necessary. I just basically wrote that you just have to, count what you can. I would say besides that, I think that everything that we tried made it into the published article. Yes.
And since the time of publication, have you encountered that the necessity for troubleshooting? Sometimes you think you have your protocol and it's running until it doesn't anymore. Mm-hmm. Have you encountered these? Have you had to troubleshoot? And if so, how?
I would say that not since it's been published. We haven't had anything, but in the process of making it, there were so many troubleshooting steps. I would say for anyone who's interested in using the protocol, one thing that you really, really, really want to make sure to do is be considerate of the bore size of your pipette tips. For two reasons. One, because if you're using cells that aren't as tough to dissociate, then you may be fine with your standard bore size. But because of epithelial cells are so hard to dissociate and get into a single cell suspension, then we had to use a wide bore tip in order to get them off of the plate initially. But then, and this can have like really negative effects downstream, but if you're not very careful about getting them back into a single cell suspension every single time after you've done the methanol acetone inactivation, then you'll get so many doublets in your single cell analysis because the methanol acetone makes the cells very sticky. And I know that a lot of the times when you're pipetting, it's like, oh, of course, get into a single cell suspension. And I'll say the difference between doing it without being super conscious of it and making sure that you're getting into that single cell suspension can literally take like hours because of all of the wash steps. So I've done the protocol in an hour and a half, and I've done the protocol and it took about 6 to 8 hours at once, which is really crazy just because of how tightly the cells were to one another. But in the end, the single cell data still came out just the same, and I will say not only can the single cell data be affected, but also it's important for your microfluidics, which a lot of the times with single cell, when you do the chips, then or basically you can clog the microfluidics. And for us, that tended to be a more expensive troubleshooting errors, because then you have to throw that whole piece away and get a new one. So yeah.
So it seems then that, dissociation and the pipette tips is a crucial step. Are there any other steps in the protocol that users have to be really, really careful with?
I would say temperatures. Temperatures, if everything isn't chilled, then you'll have lower quality RNA after a while. I believe in the published article, then I showed different time points for the inactivation, which it seemed like there's a lot of room for error there. But I would also say making sure that you have a really good RNase inhibitors.
Okay.
And high quality RNase inhibitors, because in the paper, then we showed if we didn't have any RNase inhibitors, then we still had pure RNA, but the RNA integrity significantly declined. So at that point, it honestly wouldn't even be worth it to pay for single cell at that point. So yes, I would say those are the most crucial steps.
And you have mentioned that one of the comments from the reviewers is about other applications of the protocol. For example, flow cytometry. Is that correct?
Yes.
Have you considered, and you said that that the solvents can interfere with the fluorophores.
Yes.
Is there anybody in your lab or yourself interested in doing any further optimization of the protocol to expand it to other applications?
To other applications, I would say, so I'm graduating hopefully in about 2 months. So I won't be doing it unless I'm doing it in a postdoc or at a job afterward. But no one has attempted to do it for other applications yet. So no one's doing flow cytometry or any like single cell proteomics or anything like that quite yet. Hopefully we'll get there. That's always funding dependent, but for now we're mostly interested in gene expression. So.
Right. And I would like us, we have dug into the details of the protocol itself. I would like us to move back and have, again, like a bigger picture sort of discussion at the end. And I wanted to hear what has been the impact of publishing the protocol and the paper for your young career as a scientist.
Yes. So the impact, wow. Well, again, this is my very first paper, and I was encouraged to try to do the entire thing by myself. Like, obviously like writing the manuscript, putting together the figures and all that, but also finding the journal and going through the submission process. And I'll say that for me, like as a learning experience, it has been so helpful, because when I'm thinking about my second and third papers, I believe that PLOS ONE's format and the way that everything is done has kind of made it easier for me to apply to other journals, because they have a really specific and long submission guidelines. I'll also say that using the protocol entry service, so the PLOS ONE and protocols.io, the collaboration, I believe that there's like a code or something for the entry service for protocols.io. And this is my first time using protocols.io as well. And I felt like it was really helpful for me to go back and forth with the editors for the protocol to help me to format it appropriately for the platform, and really think about like what exactly was needed. And everything looks different when it's just on a template that you use within your lab, but putting in a different way that's more standardized is more helpful for everybody. But also is really helpful for me to think about the way that things were ordered in the protocol. So I actually really enjoyed it. I felt like it was really helpful for me moving forward when I'm submitting papers and thinking about the way that I want to share my methods.
And did you like the final version of the protocol?
Yes. Yes. I would say that it's pretty detailed, but I really love it.
And so from the time that you thought it could be a good idea to submit the protocol as a paper, or you were encouraged to do so, to the time that you click the submit button, how long did it take you?
Oh,
you remember?
Quite a while, because I was also doing the doing the analysis for the single cell data that we collected. So a lot happened, but I would say if I had to like realistically about how long it took me, I would say a good 2 months after I had everything gathered, just again, because I was learning for the first time how to submit a paper, collecting, who funded me where and what time, so things like that. But I would say if I had another protocol to do, I would definitely do the same. I would definitely go through protocols.io and PLOS ONE.
Okay, good to hear.
Yeah.
And just before we wrap up, is there anything else that we haven't discussed that you would like to mention?
I don't know. I just really like the idea of having the peer-reviewed article as well as, a standardized protocol. So for me, I work in both bioinformatics and in immunology. And I actually, I've never taken a bioinformatics course. I've never taken any coding courses, but it's really easy to learn step by step how to do things online. There's so many tools for bioinformatics to learn these things. And for like wet bench work, it's not the case. There's a lot of trial and error, and hopefully you hear something from a lab mate or a neighboring lab. But I really like the protocols.io platform is so useful. I've found myself since I've published on the platform, then I'll go through other protocols, and I'm like finding ways like, oh my goodness, like I've been trying to figure out how to optimize this qPCR forever, and so I'm just really thankful that the platform exists. And with the collaboration with PLOS ONE, that it makes it a lot more searchable. I don't know. I just think that it's brilliant.
Yeah, more discoverable, right?
Yes,
more
absolutely.
searchable, more discoverable, and and also readers might feel more confident that whatever you're posting has been validated.
Absolutely.
Because it has been seen by your colleagues, your peer reviewers.
Mm-hmm. Absolutely. And also people are used to like looking at the format of a peer-reviewed paper. And so once you go into a new platform, like, so typically for me, it'd be like, like GitHub or something like that. And then now there's protocols.io where, you have these steps, you have to get used to this new format, and you don't want to invest yourself into getting used to a new format. But if you have this other, regarded journal where it's peer reviewed, you can say, okay, well, this is worth my time, so I don't know. I really appreciate the collaboration.
Well, thank you very much for your kind words. I don't want to take any more of your time. I know you're super busy preparing graduation. Congratulations again, and we'll stay in touch.
All right, thank you so much.